Hello, I'm Elena, and I'll be talking about Staphylococcus strains on phones. This is my phone. You probably have one too. Now, don't go telling everyone, but I kind of use my phone everywhere. Everywhere. And we know that all surfaces have germs. Some germs can be opportunistic, so capable of causing disease. For example, Staphylococcus is a genus of bacteria that is mostly harmless and completely natural to be found on us and surfaces around us, but some of its genetic varieties, like Staphylococcus aureus, is tough to treat when resistant to antibiotics. I sought to catalog the types of Staphylococcus strains found on phone screens and to test if they are resistant to Ampicillin, which is the antibiotic that I used. To do this, I first swabbed samples of 20 different phone screens. These samples were suspended in buffer liquid and then spread on two different types of plates. One had selective growth medium on it, which is nutrients that is especially supportive to Staphylococcus growth, and the other plates had growth medium that was infused with ampicillin. These were incubated at 37 degrees Celsius for overnight growth. And then I did some tests. The coagulase test, for example, shows if coagulase is present and helps distinguish between different Staphylococcus strains, and since some strains produce coagulase and others don't. The Kirby-Bauer test measures the susceptibility of strains to ampicillin by placing a drop of ampicillin in the middle of the plate and measuring the area around it that didn't show growth. After these procedures, I counted colonies on both plate types. There was no significant difference between the number of colonies grown on regular growth medium plates and number of colonies grown on ampicillin-infused growth medium plates. So, no clear resistance to this antibiotic. The phenotypic testing revealed that 11 of the 20 strains could potentially be S. aureus, and this is key because strains of S. aureus that show antibiotic resistance are typically tough to treat. So, overall, Staphylococcus strains were cultured, and some were likely to be S. aureus strains. To confirm this, there definitely have to be more tests necessary. The next step for this project could be to sequence DNA and run it through an online comparison program, such as BLAST, to confirm and compare each strain. Also, increasing the ampicillin dose would be useful in establishing if and quantifying how much antibiotic resistance or sensitivity there actually is, to cross-reference that with the pre-established Staphylococcus strains and compare them. This definitely illustrated the potential my phone has to carry bacteria. And whilst it's super cool to see this in the lab, I'd rather keep my phone clean.